incredible students have contributed to this topic, including Oscar. But today I will be mainly focusing in the new result of one very talented PhD student who just finished his PhD last year and moved to Curubila's lab now. And he has been unable to, to really start his postdoc for all the situation, but I'm sure he will be doing a great job there and will contribute to Reggie's lab. So today I will focus in BDNF and the regulation of neuronal morphology by BDNF and its receptors. So we know that extracellular cues as BDNF and its receptors to allow neuronal morphology have to regulate several cellular processes, such as the activation of specific transcription factors, cytoskeleton elements, local translation of proteins, organelle dynamics, and membrane trafficking. And we will be focusing a little bit on membrane trafficking. So the effects of BDNF are really well illustrated in the cortical, in the cortex, in the layer 2-3, for example, of the cortex. BDNF and its receptor is required for the maintenance of the dendritic arbors. And there are some key experiments showing that in conditional knockout mice. So if you can follow my pointer, these are conditional knockout mice by BDNF, and you can see here a wild-type neurons, and here is a knockout neurons, and you can see how there is a reduction in dendrites, and also a very important paper showed that deletion of the TrkB receptor in cortical neurons induces a complete retraction of the dendritic arbor followed by neurodegeneration. So BDNF and its receptor TrkB is very important for the maintenance of dendritic morphology in several CNS neurons. So BDNF binds two types of receptors. The P75 receptor, which is a common receptor for most of neurotrophins, and the tyrosine kinase receptor TrkB, which will trigger specific signaling pathways. And today I will be talking about two specific signaling pathways mainly, the ERK kinase activation and the P3 mTOR activation. So in central neurons and according to our experience, TrkB regulates the neuronal morphology mainly by activation P3, mainly by activation of ERK and the activation of the transcription factor CREB, and also by activating the translation of protein through the P3 kinase mTOR pathway. So BDNF then, through its TrkB receptor, is able to regulate both transcription and translation of a specific subset of proteins to allow neuronal plasticity. So one interesting aspect of neurons is that they have a complex morphology. If you see here, this is a pyramidal neuron that is compared to epithelial cell. And what you can observe is that the plasma membrane of this neuron is large, complex. It can cover a long distance, and we all know it's also very dynamic. It changes with experience, it changes with extracellular cues, and it changes according to the connectivity that this neuron has. So, for instance, and for you see how distance. The distance that a neuron can cover in the central nervous system. Here we have injected an adenovirus, associated adenovirus in the cortex, driving the expression of GFP. And you can see how all neurons are labeled, their dendritic arbor that is illustrated here in a close-up, and you can see all the axons that are projecting through the cortico-callosal pathway to the contralateral hemisphere to innervate neurons in the other side of the brain. So you can see that also in the central nervous system, the axons of neurons can cover large distances. So as many labs in the field have addressed this problem of how neurons that are responding to neurotrophins in different places, like in distal dendrites, in proximal dendrites, and in the axons, how the neuron integrates this signaling to regulate transcription and translation of proteins. So then, which is the mechanism that neurotrophins follow to allow this integration? So many labs in the field, and I'm not there to name all of them, so I don't forget, have done many contributions, and it's quite established that when a neurotrophin receptor binds its neurotrophin in the plasma membrane, initiate the signal in the plasma membrane, but then it gets internalized, and in endosomes, the neurotrophin receptor continues signaling. And now we know that they can continue signaling in different types of endosomes, and will use different platforms for signal locally or for long distance signaling. So one important aspect of one group of proteins that are important for the regulation of the endosomal system are the RAB monomeric GTPases. When neurotrophins bind the receptors in the plasma membrane, they are internalized into an early endosome that is regulated by the RAB5 GTPase. And RAB5 then allows the homotypic fusion of these endosomes that are produced upon internalization. And from there, from this endosome that's called also the sorting endosomes, receptor can move to the recycling pathway and being close to the nucleus or come back to the plasma membrane. And the times that the amount of time that the receptors can continue recycling here will regulate the availability of receptor in the plasma membrane, and will also regulate the long lasting of neurotrophin signaling. Also, how long it takes to reach to the lysosomal degradation will also be important for neurotrophin signaling. So here we have two important GTPases, RAB5 regulating early endosomes and RAB11 regulating the recycling pathway. Also, this signaling endosomes has been shown to be important to transmit long distance signaling in mainly, and this has been mainly done by many labs, in peripheral neurons where neurotrophins are secreted by the targets of the neurons. They bind the receptors, they get internalized in these signaling endosomes, and these signaling endosomes are transported back to the cell body using the molecular motor DNA. So we had contributed to several papers, or we have contributed in the lab with several articles showing that neurotrophins can regulate endosomal pathway through the activity of RAB GTPases, and this impacts. There is an impact of the activity of RAB GTPase in neuronal morphology. So one of the first paper we published, according to that, was showing that BDNF and TrkB was able to regulate the recycling pathway mainly in dendrites, allowing an increase in the recycling of receptors in the dendrites, and this was required to induce dendritic branching. More recently, we have shown that when neurotrophins bind TrkB, they enter the endocytic pathway and regulate the activity of RAB5 GTPase. And this process is also important for regulating neuronal morphology. Also, more recently, we have shown that the early and the recycling pathway is not just important for local signaling of receptor, but also for nuclear signaling. And you can find the result of this research in this, it is published already as a preprint and will be soon published in Journal of Neuroscience. And here, importantly, we show that the recycling pathway is not also required for local signaling in dendrites, but also for nuclear signaling. So activation of the ERK and CREB is regulated by the activity of RAB11 GTPase and allowing dendritic growth. So with all the years of research we have in the lab, we think that endocytic signaling and transport of these signaling endosomes is a key process that allow neurotrophic signaling integration and also transcriptional regulation. So when I think about this process, I have this drawing in my head, like there's like different endosomes reaching the cell body from different region of the neurons. So here you have the dendrites will use mainly the early endosomal pathway to arrive to the cell body. The endosomes that are generated in the axons will use specific endocytic carriers that have been are diverse in the literature, but have a common, the RAB7 GTPase will also reach to the cell body, and somehow the nucleus have to integrate this signaling. So, and we think that the endosomal pathway has a key process in this integration. So, specifically today, I will be talking about the Guillermo's work related with evidence that show that there is a functional role for a long-distance signaling in central neurons for regulate dendritic plasticity. So what we asked together with Guillermo if neurotrophins activate receptors in the axons, will be able to this signal to be transmitted to cell body to increase dendritic branch. So it will be in a cortical neuron, right? Will be like that. Neurotrophin secreted here will have to move in vivo, right, up to the other side of the brain to induce plastic changes. So to start answering this question, we use an in vitro approximation, and we use microfluidic chambers that you probably are aware a lot of them and use them normally in your lab. So these chambers allow to cultivate neurons in two different compartments. So in one compartment, you have the cell bodies and the dendrites, which are illustrated here in green. And there is a 400 microgroup that separates distal axons that are here labeled in red. Okay, so it's already well demonstrated in central neurons that BDNF, this is fluorescently labeled BDNF, is able to be transported retroclally to the cell body in a biased form. So, and here is the chemogram showing that. So we have really good evidence, together with other evidence available in the literature, that signaling endosome might reach the cell body and will trigger dendritic plasticity there. So in peripheral neurons, this pathway has been shown to mainly regulate survival and also connectivity with the next upper neurons. So we follow, we have two models, two cellular models, and we follow more or less this experimental design. So we have the cultures in a compartmentalized cultures. Here is in blue is the cell body compartment, in green here is the neuron, and in yellow you see the axonal compartment. So the flux of the fluid is always going, depends in the experimental design, but mainly is going through the axonal compartment. So what you add here will not be able to reach the cell body compartment. It will just signal, or will just have access to the axon. And because BDNF is secreted from the in dendrites and also in axons, we quench the BDNF that is secreted in the axona, in the cell body compartment, with a chimeric protein that is a soluble TrkB receptor. So even if we have a little bit of flux in this direction, it will be trapped by the TrkB Fc chimera, and also the secretion of BDNF here will be quenched. We use two cellular models. We use cortical neurons, and we use the classic K252A kinase inhibitor to inhibit the activity of TrkB. And we use cortical neurons derived of this knocking mouse, the FC16A knocking mouse, that it has a mutation in the tyrosine kinase domain, activity domain of TrkB, and this allows that this mutant receptor will be sensitive to this molecule and NMPP1 in a specific manner. So it will allow us to add the inhibitor in different compartments and study the influence of inhibition of TrkB in different sites of the neuron. So the cultures look a bit like that. These are neurons that has been transfected with GFP, and GFP will mainly label the dendritic compartment and also a little bit the axons. And we distinguish then the neurons that are labeled with MAP2 that will be labeling the dendritic morphology and GFP. And also to analyze neurons that we know have axons in the axonal compartment and therefore are responding to the drugs that we add here in the axon. We label with a retrograde tracer that's called cholerotoxin and is fluorescently labeled. So you add here the cholerotoxin and it will be retrogradely transported and will be accumulated in the cell body. And you can see these pink dots there. So this is the cholerotoxin that has been transported to the cell body. So then we ask, we have this experimental design, if you follow my pointer here. So we have BDNF in the absence or presence of a tyrosine kinase inhibitor here to study whether it's able to induce an increase in dendritic morphology, and also whether the activity of these receptors are important for this process. So what you have here is that when you add BDNF to axons, so here in the yellow, and the signal has to move up to the cell body, you increase the morphology, the dendrites of the neurons, and you can see this here. But if you have the inhibitor of the activity of the receptors in the axons, this effect is here abolished. So this is true for the number of primary dendrites and also for the branching points, and also when you analyze the branching points and the neuronal morphology using here a Scholl analysis. So very similar results we obtain when we use the mouse neurons, and we add the NMPP1 inhibitor in the axonal compartment. So we also obtain an increase in dendritic morphology upon addition of BDNF, and this was reduced by the inhibition of TrkB activity in the axons. So we also wanted to know what was the requirement of P75 expression in this signaling event. So we did the same experiment using neurons that were or wild type with full expression of P75, heterozygous mutants, so half of the expression of P75 or completely knocked down of P75 using the exon 3 mouse knockout. And we could see that wild-type neurons respond to BDNF and also knockout neurons. So the expression of P75 was not required for the response of BDNF added to axons. However, if you follow me here in this part of the presentation, when we analyze the neuronal morphology of different phenotypes, genotypes, and we could see that the basal levels of neuronal morphology were different in wild-type neurons, that is the blue line, compared to heterozygous and knockout mice, which is the red and the black line. So what you can see is that knockout neurons have less primary dendrites and early dendritic arborization, but have an increase in ramification of distal dendrites. So this means that although BDNF is able to signal in a P75 knockout background, P75 will be important probably in vivo for the full morphology of to a physiological, for the neuron to have a physiological morphology. So we know that, as I mentioned in the introduction, that the CMP response element binding protein CREB is important for neurotrophin signaling, but is also a classic transcription factor activated by neuronal activity. And it triggers the activation of immediate early genes, and between them is BDNF. So BDNF, CREB will activate the transcription of BDNF, but also BDNF will activate CREB. So for the maintenance of the dendritic arborization, we think there is a bidirectional loop of CREB and BDNF activation. So we think that CREB has to be also relevant for neuronal morphology when BDNF is signaling from axons. So to study this, we did some experiments in vivo. These are preliminary data, but just to show that the phenotype of decreasing the CREB activity in pyramidal neuron is similar to decrease the activity of TrkB. When we express a dominant negative of CREB in the layer 2-3 of the cortex, here with adeno-associated virus, you can see here the morphology of a pyramidal neuron that is expressing mCherry and GFP. And here you can see the morphology of a neuron that is expressing the dominant negative of CREB and mCherry. So here is the CREB in the nucleus, and here is mCherry. So the dominant negative of CREB also here is better illustrated the change in morphology. So we have to reproduce this experiment several times more, but it's very suggestive that expression of a dominant negative of CREB will have a similar phenotype that reducing BDNF and TrkB. So then we ask whether CREB is activated by axonal BDNF and whether CREB activation is required for dendritic branching inducing by axonal BDNF. So to do this, Guillermo followed an experimental design where he labeled overnight the cells that have axons in the axonal compartment with one color of the cholera toxin that is red, but add another color of cholera toxin when he added BDNF. And he added BDNF for different times, from 30 minutes to almost 3 hours. So, and here you can see how CREB is getting activated. CREB is here in green with the addition of BDNF. So you have a rapid activation of 30 minutes, and then the activity is down-regulated, but is up again after 3 hours. So we have a biphasic activation of CREB upon addition of BDNF. Interestingly, if you reduce the activity of dynein, which is this motor that will transport a signaling complex from axons to the cell body, you inhibit the activity of dynein in the axons with this drug that is ciliobrevin. You will reduce the activation. Here is shown the activation of CREB by BDNF almost to its basal levels. But if you perform the same experiment when you stimulate for 30 minutes, you will reduce the activation of CREB, but still neurons are able to respond. So there is a reduction if you inhibit dynein, but not a complete reduction, suggesting that at early time points there are other components that might be also important for long distance signaling. So then we ask whether the addition of dynein in the axons or the inhibition of CREB in the cell body, but this drug that will reduce the binding of a coactivator of CREB, that is CBP, are able to diminish neuronal, the dendritic morphology induced by BDNF in axons. And here I am showing you the result of the inhibition of CREB in the cell bodies when BDNF is added to axons. So here is the induction of dendrites by BDNF, and there is the diminution of dendritic morphology if CREB is reduced. And also, you can see here in this CHOL analysis, if ciliobrevin is added to axon, the effect of BDNF is reduced. So dynein reduces the activation of CREB, and CREB is then required for the effect of in morphology of BDNF added to axons. So another question that we asked was whether, or we wanted to know, so we know that the activity of TrkB when you add BDNF in axon is important in the axons. So we wanted to know whether the maintaining of TrkB activity is required in the cell body when BDNF is added to axons. Because you theoretically can think in two ways that the signal is transmitted from axons to cell body. One is through signaling endosomes, that is the hypothesis we have, but also it's possible that TrkB in axons activated a signaling complex that has, for instance, active ERK, and this complex is transported to the cell body. Such a mechanism has been shown for peripheral neurons by Eran Persson and Mike Fansilver some years ago. So we wanted then to know whether the activity of TrkB in cell body was required. So first, we ask whether we can find active TrkB in the cell body when we have stimulated TrkB from axons. For that, we transfected neurons with a epitope target TrkB that has the FLAC epitope in the amino terminus. We transfected neuron with this plasmid, and then we label the TrkB that is in the surface at 4 degrees with a FLAC antibody. Then we added BDNF for several minutes. Normally, I think these experiments are done with 3 hours of BDNF. We fixed neurons, and we did an immunostaining for phospho-TrkB. So in red, you will see the FLAC TrkB, and in green, you will see the phospho-TrkB. So what you see here is a sum of FLAC TrkB and phospho-TrkB that is in proximal axons, so very close to the cell body. So, and what you can see is that there is a colocalization of TrkB FLAC with active TrkB in several of these axons. And also, you can see that there is an accumulation of active TrkB in neuronal cell bodies, suggesting then that you have an active TrkB that is being transported from axons up to the cell body, and this is required to trigger signaling in cell bodies. So to show that with a different experimental design, we use the mouse neurons, the knock-in mouse neurons that we have, and that respond to the MMPP1 inhibitor. So, and the experiment we wanted to do is to add BDNF in axons, activate TrkB in the axons, and reduce its activity upon arrival to the cell body. So what happened if we do that? But first, to know if this experiment was possible, we needed to know whether the NMPP1 reduces the activity of TrkB once the receptor is activated. So first, we did this experiment So you have in non-compartmentalized neurons where you have the control, so no BDNF. Then you have serum depletion, and then BDNF for 30 minutes. You wash, and you see how long it gets activated, up to 30 minutes more. Then you pre-incubated with 1 NPP1, and then you add BDNF in the presence of the inhibitor. So this is the negative control. And the one we wanted to compare is that we just do serum depletion here. We add BDNF for 30 minutes, so the receptor is activated, and then we add for 30 minutes more the inhibitor. So when you do that, and the lines you want to compare is the line 2, this line, and the line 2 with the line 4. And here you can see the full activation of TrkB with this experimental design. So it has seen the BDNF for 30 minutes, and then we have removed it. And then if we have added, and here is when you have 30 minutes of inhibition. So here we can see an inhibition. So here we have quantified this, so it's activated. And if it has the presence of this last 30 minutes, an MPP is reduced to basal levels. This is more easy to see when you see the activation of AKT. So here you see the activation of AKT after 30 minutes of BDNF, and how this is reduced if you treat the neurons with an MPP1. So these results, and we did this for other downstream signaling pathways, such as the mTOR signaling pathway, and also CREB. So I'm not shown here for time reasons. So this suggests that when the receptor is active, you can reduce the activity by adding an MPP1. So probably the receptor is getting active and inactive all the time, and in this moment, an MPP1 is able to reduce the activity of the receptor. So this means that we could follow this experimental design and trust our experiment. So this is what we did. So this is the normal situation, the control, nothing, just a cholera toxin adding to axons. All the cultures have cholera toxin. One will have BDNF added to axons, and with the addition or not of the inhibitor in the cell bodies, and as a control, we use inhibition of TrkB activity in the axons. And here, what you are following is the activation of TrkB in the cell bodies. So when you add BDNF in the cell bodies, you can see, in the axons, you can see an increase in phospho-TrkB in the cell bodies, and this is abolished by the presence of an MPP1. So suggesting that upon arrival to the cell body, in MPP1 in cell bodies will be able to reduce the activity of TrkB, and this is parallel by the activation of TrkB. So here in green is CREB, by the activation of CREB, sorry. So here you see before you were seeing in green the activation of TrkB, and now you are seeing in green the activation of CREB, which will be the downstream effector and is required for dendritic morphology. In red is the cholera toxin, so you know that these neurons have seen BDNF in the axons, and in blue is the nucleus. So when you add BDNF, as I showed you before, CREB is active, but if you reduce the activity of TrkB in the cell body, then the activity of CREB is also reduced. So all these results suggest that the activity of TrkB is required in axons, and is also required in the cell body to allow transcriptional regulation. So as I mentioned in the introduction, and these are the final slides, when BDNF binds TrkB, it activates the CREB pathway through ERK, and also activates the PI3 kinase pathway and the translation of proteins. So we wanted to know whether both processes were upregulated in the cell body when we added BDNF in axons, and whether they are co-regulated, so both are required for final protein translation. So for that, we studied activation of mTOR targets. This is, so the ones we chose because they have nice antibodies that work for immunofluorescence is the activation of phospho4-EBP1. And you can see that if 4-EBP1 is get active in a time-dependent manner when you add BDNF in axons, getting a peak at after 3 hours. And here is the kinetic of activation and phospho4-EBP1, or the kinetic of phosphorylation of this protein, sorry. And also another target is activated, and this target is the S6 ribosomal protein. So 2 targets of the mTOR pathway are activated in cell bodies when BDNF is added to axons. And this, just to show you the specificity of this experiment, so when you add BDNF to axons, 4-EBP1 gets phosphorylated in the cell body, and this is reduced if you add in axons ciliobrevin that will reduce dynein activity, so you need a transport process, and also will be reduced if you inhibit P3 kinase and the mTOR pathway in the cell bodies. And this is quantified here. So finally, we wanted to know whether BDNF added to axons will increase protein translation in the cell bodies. And to answer this question, we used the click chemistry that uses an amino acid that is modified, and then you can label it with a fluorescent tag. And then you can see how this modified amino acid is newly incorporated in protein. So here, when you add BDNF to axons, you can see that there is so this is the control situation, and we use as a readout an early gene, the expression of an early gene that is required for the dendritic and synaptic plasticity, that is ARC. So when this is the control situation, and when we added BDNF, we could see an increase in the AHA incorporation. So there is indicating that this there is an increase in protein synthesis, and this is parallel, but the increase in the synthesis of ARC. So finally, we wanted to know, to give a specificity to this process, if this increase in protein translation and ARC expression was reduced by inhibition of the CREB transcription factor that will be shown here in the presence of BDNF in axon, and or the inhibition of the mTOR pathway that will be shown here. So what you can see here for the levels of the AHA incorporation in neurons, when BDNF is added to axons, there is an increase in incorporation of AHA in the neurons, and this is reduced by both, but by the inhibition of CREB and by the inhibition of mTOR. And this is, and the same results are where we found the same result for the ARC levels. So these results indicate that BDNF signaling in axons can coordinate the activation of transcription and translation to increase protein synthesis in a specific manner. And a matter of future research will be to try to understand which are these proteins that are getting synthesized specifically by BDNF added to axons. So here, as we know, we like models to try to see this in a more simplified manner. So we think that when BDNF are activating TrkB in axons, the receptor gets internalized, and then the endocytic pathway transports this signaling complex up to the cell body. And the TrkB active here in the cell body will turn on CREB and mTOR, and there will be a specific mechanism that we don't know which mechanism is, that is able to coordinate both processes, such as that CREB will increase the transcription of the transcript that are actually being targeted by the mTOR. So finally, I think our results shed lights on whether there is a signaling role in wiring neuronal circuits in the CNS. And we think that we have given in vitro evidence, and of course we will have to develop this further to give in vivo evidence that when axons in the CNS see neurotrophins, it triggers a signaling pathway that can reach the cell body and regulate neuronal morphology. So, of course, I want to acknowledge the people who have done this work, Guillermo again, Luisa, and Nicolas, and past members that have done a great job in developing these projects through the years, Oscar, Andres, Carlos, and the projects that funds me, and of course the collaborators in these projects, Eran Persson, William Mobley, and Chenbia Wu, and Gipi Schiavo in UCL. So thank you very much for being here, and I'm ready to take questions. Oops. Okay.
Many thanks for your presentation, Kika. Really, really interesting. We already have a question in the chat from GP Schiavo, and I don't know, GP, if you want to, GP's next door, by the way, if you want to make the question yourself.
Voice, no.
Yeah, no, please.
Go ahead.
Okay, so I will read it out from the chat. He obviously say thank Kika for the nice talk, but he asked whether or not the reduction of CREB activity by using the AAV could be causing a global change in the size of cells instead of altering branching and morphology, as he has seen by reducing global concentration of main motor complexes, for example.
No, I don't think you can exclude this possibility, because when you remove, for instance, TrkB, you also see the same. So first you see a collapse of the dendritic arborization, but then you will see a completely atrophy of the neuronal cell bodies. So, well, I think that TrkB and the pathways that TrkB regulates are really important for the maintenance of the neuronal viability at the long run. But the thing that we can learn from that is that connectivity, as we obviously know, is very important. So first a neuron will get disconnected and then the neuron will degenerate. And I think this is a very well-known process in many neurodegenerative diseases.
Can I ask a question?
Please.
So Kika, that was a beautiful talk, as always. I was curious, connecting the TrkB activation with CREB activation. So is it through the ERK pathway that the TrkB is activating phospho-CREB? Is there a role also for other signaling pathways? Does AKT play a role? Can you talk a little bit about, sort of how you get from TrkB to CREB and what signaling pathways you think are involved?
Yeah. So in this paper that I mentioned at the beginning, that it will be out soon, there, and it's in a preprint form already published. So there, but this is not specifically on BDNF added to axons. So this is not long distance signaling. But there we show that it's the ERK pathway that is activating ERK in central neurons. We don't see an effect of the P3 kinase pathway in our model. So these are hippocampal neurons that are in a plate. So here in the model of long distance signaling, we have studied the PI3 kinase pathway. We know that activation of PI3 kinase doesn't have a role in the axons for transport, but obviously have a role because it's activated the mTOR pathway, and the mTOR pathway and local translation of proteins is important for neuronal morphology, but it's not. But in what we think is that we will have to, we didn't add a PA3 kinase inhibitor and see whether CREB is phosphorylated in this model. But there's some preliminary results that Guillermo also has that is interesting, is that, for instance, activation of phospholipase C is not required for CREB signaling. So in this model of long-distance signaling, we really don't know, and we will have to study which is the kinase that is activated in the cell body to activate CREB, but I think this will be probably ERK, so if I have to guess.
Thanks, Kika.
I have a question. Hello, Kika.
Hi.
Nice talk. I have two questions. So first, with the days in vitro of your cultures, I was wondering if there is a difference, and if you did this experiment in different days in vitro, like more mature neurons, and if the BDNF signaling or effect in morphology will be different, or you don't expect any difference. And my second question is regarding with the signaling too, you showed in your blood the increment in the phosphorylation in serine 473 of the AKT, and I was wondering if you look for the phosphorylation 308, because we know in the literature that the 308 phosphorylation in AKT is required for the activation, and the 473 is like modulating the function. So I was wondering if you see also an increment in the phosphorylation of 308 too in the AKT.
Oh, so yeah, no, in TrkB I just forgot the days in vitro.
Yeah.
So I think the days in vitro we use is 6 days in vitro, and then we add BDNF for two days. So neurons are normally not older than 9 days, up to 10 days. If Guillermo wants to add something, he's welcome. So we haven't analyzed older neurons. But something that I didn't mention, because it's a detail in the protocol, but it's important, is that because, well, one thing you would like to know is whether this BDNF axonal signaling is able to increase, for instance, synaptic synapses, the number of synapses as well. So we tried to do some experiment in this direction, but it was not so easy to keep the neurons so long. And also, if you don't block BDNF in cell bodies, you won't see an effect of axonal BDNF. So I don't think it will be, I don't know. I think if you want to study synapses and synaptic physiology, we really have to think in a way to see this in vivo and go for an in vivo thing. So I think we have that. So we have done a proof of concept experiment. So in the sense that BDNF from axons in central neuron is able to regulate plasticity, and I think the next thing would be try to see this in vivo.
Okay.
Francesca.
Please, Moses. Yeah.
I enjoyed your talk. CREB is activated by co-activators. There's one called CRTC. And it's a very interesting protein because it shuttles back and forth into the nucleus. A lot of work has been done by Kelsey Martin and also Mark Mondment. I think it'd be interesting to test what is going on with CRTC in your microfluidic experiment. It'd be very easy to do. They're very good antibodies against the protein, and it's regulated by many signals. So you'd be able to see, what is actually activating CREB in the nucleus.
Yeah, thank you a lot for your suggestion. I just took note of that.
I'll send you a paper.
Yeah, great. So Reggie is asking if enhanced translation is restricted to the soma or it goes locally in dendrites. Yeah, that's a great question. And I think we will have to really answer these questions we will have to use specific tools to see local translation in dendrites when BDNF is added to axons. But what we saw is that this early gene, its mRNA is translated to dendrites, and we saw immunostaining of ARC in dendrites as well. So this suggests that I think maybe these mRNAs are being translated in dendrites as well, transported and translated in dendrites as well. I think they have full of time because they have 3 hours to do so. So I think it's possible, but we didn't show it.
Kika, I have a question.
Hi, Christopher.
So something that we found a while ago but never published was that, at least for the NGF-TrkA signaling endosome, MAP kinase, like, so if it's originating at the distal tips of axons, and then it goes back to the cell body, for synapse formation, it would have to go into the dendrite. And what we found was that MAP kinase was required for the signaling endosome to go into the dendrite. So I was wondering if you had actually seen, if you have ever looked at whether inhibiting MAP kinase also inhibits TrkB trafficking into the dendrite, which could then potentially influence translation in the dendrite and also dendritic elaboration. Yeah,
no, we haven't done this experiment, but yeah, it will be a great experiment to do. These are the things when you have this model, and this is the nice thing of Guillermo's research is open. It has a model. There is a cellular model to study many possibilities now. Like if a signaling endosome generated in the cell body, an axon reaches cell body, and in addition goes to other places in the neuron to if they tag some synapses. It will be great thinking that yeah, from such a long distance can influence what is happening. But this you already yeah, in this paper that was published by the Gintis lab. I don't remember who you were there. It suggests that, right? You were there, probably.
Okay. Thanks, Kika.
Kika, if I may, I would like to have a follow up on the question from Christopher, because can you comment a bit more on the results you show on the P75 knockout? Because this appears not to be required for primary dendrites, but does it for branching? And the other thing is that you see these basal differences on the morphology of the knockout versus the wild type. So how do you interpret that? Do you attribute that to maybe a difference on the sorting or trafficking of signaling endosomes from axon to distal dendrites? Maybe P75 maybe with TrkB that requires MAP kinases to get there, for example.
Yeah, that's a nice possibility. So overall, we did all this analysis. Guillermo did all this analysis: primary dendrites, branching, and there's no difference. So neurons are able to branch and are able to branch primary dendrites, and there's this difference in basal morphology. But BDNF is able in the knockout to increase this, right? So it's hard to see then to say that the P75 in this model have a role. Also, we saw that CREB activation doesn't get influenced by the presence of P75. But in vivo, I think the result will be quite different, and it's very possible that P75 has a role. Yeah.
Sorry, Kiki, if I can just follow up on that. You know, we were looking at the interaction of P75 and TrkB with BDNF responses, and what we had found was that in the P75 knockouts, BDNF had the ability to activate AKT was diminished, but there was no effect on the ability to activate ERK. And so that's in line with your suggestion that is the ERK pathway that's needed to phosphorylate CREB that's involved in this dendritic morphology, and potentially that P75 is not required for that particular pathway. But it has effects, of course, in other aspects of BDNF signaling, which, of course, would be important in vivo as well.
Yeah, it will be then interesting to analyze local translation of proteins. So maybe there is a subset of mRNAs that require P75 to be translated, or not.
That is very interesting with the result, the comment, Dr. Friedman. I am seeing something similar, the ERG activation, but not AKT in mature neurons. So that's why was my question also for Kika. That is very interesting.
By the way, I didn't answer this question. I think we just analyzed the activation of these tyrosines. So it will be interesting to analyze the activation of other tyrosines. So you're right.
Good. Do we have any other question from the audience? All right. If not, we give a final applause to Kika, and thank you very much again.
Thank you, and applause to you that are here.
Thank you.
Thank you a lot.
Thanks, Kika.
Thank you all for being here today.
Thanks all.
And see you on the 10th of September, but for the presentation of Subhashini Joshi from Newark, USA, talking about P75 on oligodendrocyte development. See you then.
See you then.
Bye. See you. Thank you so much, Kika.
Bye.
Thanks, Kika.